So the schedule today for our MorphoBank workshop, so we anticipate it'll be about maybe an hour and a half, an hour and 45 minutes. We have the welcome and I'll do an introduction to the background of MorphoBank. And then Tanya will show you a couple projects and highlight the different features of MorphoBank. Then we'll take a short break and then we will have a project tutorial where I will show you the process of adding data to MorphoBank through an example of if your paper is already published and you want your data into MorphoBank, the simplest way to do that. And I think after we do that, we'll take another break. And then if anyone wants to join us for the 2 hours, we'll have a project office hours and you can bring either something you're working on, something maybe that you've already published, and we can help you get it into the bank. Or if you've already started something and you have questions, we're happy to answer that. Okay, so a little bit about myself. I have been a data curation scientist at MorphoBank for the last 3 years. I have a PhD and a master's degree in paleontology from South Dakota School of Mines and Technology. This is in Rapid City, South Dakota. I have a bachelor's degree in geology from Texas Christian University. And my research spans a couple different things, mainly conservation paleobiology and ecology. I study different river processes and how it affects food webs and any kind of nutrient contamination. I also do other kinds of work with open and fair data. That's some of my MorphoBank passion and equitable science.
And I'm Tanya Berardini. I'm the director of MorphoBank, and I'm the chief scientific officer at Phoenix Bioinformatics. I've been with the MorphoBank project since 2018, and in 2021, MorphoBank was transferred to Phoenix Bioinformatics from its original home for 20 plus years at Stony Brook University, where it was started. I have a PhD in biology from Boston University. I did my undergrad at the University of the Philippines, and my interests are in digital research resources and their sustainability, in promoting data sharing and community building. My other project that I am involved in is the Arabidopsis information resource, and that is a plant genomics database that is used by plant biologists all over the world.
Thank you, Tanya. Okay. So a little bit of a background into MorphoBank, and you probably got some of this if you attended yesterday, but there'll be some new information here as well. So MorphoBank is an online database and a matrix building website for evolutionary researchers and phylogenetics, and specifically systematics and cladistics. But MorphoBank can be used for a wide range of data, and we'll kind of go over some of the options. As Tanya mentioned, MorphoBank was launched over 20 years ago, actually close to 25 years ago now. It launched in 1999 at Stony Brook University under Maureen O'Leary, who gave our keynote yesterday. It was transferred in 2021 to Phoenix Bioinformatics, which is a nonprofit, and that is where it's been housed since and doing well. So MorphoBank data is downloaded by a variety of people, including the public, researchers, and students. It is used for educational purposes, along as it's used for additional research. So once you publish your project, anyone can access it. It can be reused for other phylogenetic studies, or it could be used for teaching and educational purposes. Data in MorphoBank is either added during the research process. So as you start off a project, you can start a MorphoBank project and add in data as you go and collaborate with other researchers around the world. You can also add your data to MorphoBank as a repository. So as you're publishing your paper with a journal, there may be a requirement to deposit the data somewhere that's accessible. And so as you're submitting your paper or it's going through the publication process, you can add your data to MorphoBank then. And then there's also the post-paper publication. So if you've already published work and you don't have your matrices in MorphoBank, you can search to see if MorphoBank already has the data, because as a curator and the team of volunteers I lead, sometimes we go in and find papers that already have this data and we add it to MorphoBank. But if you don't see your data in, you can always upload it. And it's a great way to share and reuse the data. MorphoBank has over 1,300 phylogenetic matrices, and this represents over 20,000 species. There are over 2,000 projects, and there is over 177,000 images. All of this data is used by over 4,000 scientists at over 1,000 institutions. And all of the data, for the most part, is associated with peer-reviewed publications. And so this represents about 200 journals. MorphoBank is open access, and all the data is fair, so findable, accessible, interoperable, and reusable. When you go to MorphoBank, you can have access to published project data. All of the published projects have permalink and citable DOIs. So you can cite the original paper where the data is from, and you can also get a specific DOI for the paper or for the data, and you can reuse that. The matrices are integrated into the database and they're reusable. We offer specialty data options, so 2D, 3D anatomical images can also be added, and you have access to curator support. So that would be Tanya or I can help you with any kind of formatting or publishing requirements that you need. And to access curator support, you go to curator, or you can email curator@morphobank.org. The data in MorphoBank is set up so that it can be used for a couple different things. One is you can input information on just a specimen and connect that specimen to taxonomic information. So the taxonomic name, the publication, repository information, different morphological information using anatomical labels, different character state information, and all of this data, it can be linked to external databases. So we have EOL, which in iDigBio, which you can get images for specimens from if you don't have images ready to upload. We're also connected with PBDB and NCBI for different taxonomy checks. With the data, you can do phylogenetic analyses in TNT, PALP, and Mr. Bayes. So previously, PALP was the one that MorphoBank had, the main analysis that MorphoBank had, but in our new beta version release, we will have additional analyses with using TNT and Mr. Bayes. All the data that you upload does become publicly available once you publish it. And so this can be information on specimens, taxa, images, matrices. Our goal is that this published data is shared. So you can discuss it in a journal article, you can create a data note, you can link it to other repositories, and our goal is to make everything reusable. Okay, MorphoBank projects are associated with a variety of data from hundreds of different sources. We have a wide range of journals, so there's over 200 journals, but the majority of our data is published in a couple of different journals, and so I list these here. So the Journal of Vertebrate Paleontology, Cladistics, Zoological Journal of the Linnaean Society, the Journal of Systematic Paleontology, and AMNH. MorphoBank data has been cited over time, and so this graph on the bottom left shows the increase in citing of data over time. And so as you post your data online, you can cite it in a paper that you're working on, or other people can reuse it and cite it. And so that's what this figure is tracking. MorphoBank is a specialized repository. So general repositories would be like Zenodo, whereas a specialized repository only takes specific types of data. For MorphoBank, our data structure is for phylogenetic matrices, and MorphoBank was specifically designed to build, curate, and house these matrices. This ensures the long-term usability and discoverability of the deposited data. everything in MorphoBank is open access, and it is supported by a couple different things. One is Phoenix Bioinformatics, another is NSF funding. NSF has funded multiple grants over years to set up MorphoBank, and now we're just ending in December our last NSF funding. Besides Phoenix Bioinformatics and U.S. National Science Foundation, we have a number of member institutions that support MorphoBank, and those are all listed here. Okay, on to projects. So we'll show off a couple projects to highlight the different capabilities that MorphoBank has.
And I will take over the sharing. Let me just make sure I can get the right page. While I look for that, if anyone has any questions, on Brooke's presentation so far, let's pause there. Just trying to see what the second project number, Brooke, is what?
Is 832. So Tanya will show you two different projects. And are you going to show in our beta version?
And we're almost there. Only had like 5 years to get used to Zoom. Okay. We have many screens open that all look the same. Oh, that's it. So please let me know if you see.
I see tear analysis. I don't think you're sharing the right screen.
Not the right screen. Let's try again. How about this? Does that look like
yes, we can
the beta
see
site?
Yes, that is the data site. We can see it.
Okay, great. So if you've used MorphoBank before, I'm just going to pull up the current site. This is what you're used to seeing. And what I thought I'd show you today is the beta site. The look is different, the speed is different, and the functionality should be the same. So I'll show you this in the browse version. Brooke will take you through in the project creation through the what's available right now on the production side. But know that once we push the new software into production, which will be probably within the next month, because right now we're soliciting feedback on the beta version, it's going to look less like this and more like the new version. So we have an overview of all of the projects that are in MorphoBank, and then you can click on them to see the details of the project. There's a search bar. You can search by the number of the project. If you're looking for a specific one, you can search by author, you can search by the organism. It's a very broad search. And I'm just going to do a search because I'm going to show you project 773 to start, but I'll show you the different categories of data that you can get information from. And you may or may not notice, but in this case, I'm not logged in. So without having to have an account, you can browse and download and view the data in MorphoBank. So it's looked for 773 in projects. There's also a search of project authors, of media, specimens, characters, taxa, and matrices. And as expected, when you're searching with a number, the result set is fairly small. And I'm going to go, actually, yeah, I'm going to go to project 773. Here it is. If you've looked at MorphoBank before, the overview page looks fairly similar. And it gives you kind of the information on the paper that it was based on, as well as the information that's in this project. So there are 12,000 some media images, one very large matrix, a whole bunch of documents, many taxa, many specimens, and many characters. So this is one of the biggest projects in MorphoBank, and it shows a lot of the features that one can avail of in the matrix editor, and that's why I picked it to be one of the ones to feature today. You can also see all of the different contributors. If you were in yesterday's session when Marina Leary was speaking, she was talking about the 25 different contributors all over that participated and added information to the project. And here's who they all are, and the different things that they worked on. It's kind of a like, wow, let's look at all of the people that contributed. And if your group is 2 people, 5 people, or 25 people, you can create and contribute data in MorphoBank. And I'm going to scroll fairly quickly. There's also a table with all of the taxa. Again, this is one of the largest projects, so the table is very big. But you can imagine if you had your matrix of 50 taxa or 300 taxa or 10 taxa, that you would have aggregate information on the project overview page. You might notice on the side that there's some information about project views, media downloads, and matrix downloads. And if you look at this number here, you might think to yourself, oh my God, that is gigantic. And it kind of is. It is still, maybe until about 2 or 3 years ago, this actually reflected people looking at the projects. As soon as the AI revolution took off and scraping started happening, a lot of this traffic reflects scraping. So we are still disentangling the people from the not people. So it is a highly viewed project, yes, but maybe not 2 million. The media downloads and the matrix downloads are more reasonable numbers, and it's not yet, the AIs haven't yet figured out how to download individually and hit at least the same monitoring process that we use for seeing how many people are accessing the data. So if that's it's something that you'd like to track, or it's something that you're like, oh, I didn't know I could look at that for projects, we're working on stripping out the bot traffic and leaving just the human traffic. But it's like this arms race. Okay, let's go into data. I've opened up each of the tabs here because I was trying to hedge my bets on like, is my internet going to go down or not? So I'll look first at the, well, actually, let's start with the media tab. So this one I told you earlier had about 12,000 different images. Looks a little bit different from production, but still, when you click on the individual image, you get a larger one. And when you click on zoom, you can get a bigger image and you can even zoom in more. So depending on the resolution of the original image that was uploaded, as you zoom in, it will get, it may get fuzzier or not. Back to media. You can look at it in a thumbnail view, which is sometimes nice. You can look at many items per page. One of the things that the new overhaul has done is to make the loading of the images much faster. We store it differently now if you're interested in how that was done. And then you can, of course, scroll through different pages. The views for the media are like in production right now. What we will add in the future is a link to the number of images and the images that are labeled as this view. So if you wanted, for instance, to see how many of or which images in this project show the medial basic cranium, you could click on that and see the images. That is not yet there. We have a list of all the specimens. If you click on them individually and they're vouchered, you get additional information on the on that species. you'll notice that there's a dagger that indicates if the species is extinct, and that's something that you can do when you're entering your data. Or sorry, if the taxon that's linked to that specimen is extinct.
For that page, can you also show the unvouchered versus vouchered?
Sure. So for unvouchered, you'll notice that there's nothing for the repository and the collection. I'll click back to this. And so you have a repository catalog number, institution code. I'll just pick another one here. This is a DMNH. So there's a variety of ways that you can specify the specimen's information. And then we have taxa. Again, 6,000 taxa is a lot for a project. So in this case, you can actually see that you can, narrow down according to the first letter of the genus. If there is a partition or if there are numerous partitions in the matrix, you can view just those taxa that are included in the different partitions. Bibliography, again, this is a huge bibliography. Usually there is one or two, and if it's one, it's the paper that is associated with this project. But in this case, we have very many publications associated. And again, you can sort them by the first letter of the last name of the first author. Finally, in the documents folder, people sometimes upload other information that are not media and not matrices, or matrices that contain, say, molecular information. Those don't go into the database. Only morphological data matrices go into the database. And so if you want to include additional information, you can upload that into the documents. You can group them into folders, or you can just upload the documents one at a time. Okay, now let's go to matrices. So this is a fairly large matrix. We've overhauled the UI a little bit to make download more clear. Viewing the matrix now opens a new tab. In the old system, it would open a new window. And sometimes that was confusing. You'd have to hunt for the matrix. But it looks very similar to the matrix editor or the matrix viewer that you're familiar with if you've been using MorphoBank before. You can tell that if there's a label associated with the image. So here's large image, one annotation. You can look at the annotations that have been made by the person who uploaded the data. You can just show names and numbers. So, and by names and numbers, I mean the name of the character state and the number of the score. Or if you want to look at it in a very condensed version, you can choose bird's eye view, and now you have something that looks like you used to looking at with maybe Mesquite or the other one, the TNT viewer. I'm not going to go into scoring because that's what Brooke might show you, probably show you after uploading a matrix, and how one could change the scores. We have tried as much as possible to test the functions of the matrix editor and viewer, but we can't think of all the use cases that all of you would use the editor. And so in the beta testing phase, we're really looking for people to go in and either in the viewing the matrix mode or in the editing the matrix mode to try those features and make sure that they're still working as expected in the new version. So I'm just going to click on a couple of things. So clicking on characters brings up the character list. One can search it with various different conditions. And if you haven't seen this before, you can highlight cells that have certain characteristics. might highlight those that have images associated and remove the highlight. So the editor and viewer has many nice features for being able to make sense of the data that goes beyond what one can do, say, in an Excel spreadsheet. Some people track data that way. Okay. Any questions about that project so far? I see a couple of things in chat. I don't know if there are questions. Brooke, are there questions?
No, no questions.
Okay. Okay. So I'm going to move
one
this.
Oh, well, now they're coming in. Has the character state label protocol changed at all? So between the beta version and this new one, I think everything labels before were a bit awkward.
So if you're the person that asked that question, please unmute yourself and then we can get to the bottom of it. Hi, Rob.
Oh, I can't hear you.
Yeah. And I'm going to pop over
Let's
to
move
the
to
view.
another question. He'll fix his audio. Someone else asked, what does NPA mean? So in here, there's some, and then in this matrix.
Oh my God, I'm blanking on what it means. Brooke, help.
I
Not,
maybe
I think it's like
instead
not,
of a question
not
mark.
yet scoreable.
I think I know.
Yes.
NPA means not presently available, if I remember correctly.
Yes, that's what it means. Exactly.
So it's sort of like to be honest, I don't remember why that couldn't stay just as a question mark. I think it was something along the lines of this, back in the day, there some character that was like within a tooth that in theory you could code, but you open up museum drawers before we could virtually dissect stuff and you'd never find the specimen. So that ended up being a not presently available instead of a question mark, I think.
Instead of a question mark. Okay, that sounds reasonable, and it kind of predates my time at MorphoBank, but thank you. But you had a question about character state
Yeah, it was
labels or labels.
That's right. So just for my own practice, because it's so labor intensive to put images for in every single cell, which is more of a reflection of my laziness rather than the best practice is to do exactly that, to document every single character state of every single coding, like what you've just shown on the screen right there. What I do as kind of a next best thing is populate that very top row where you can see starting 1, it says Gen skull telescoping exactly. And just as you hover over those cells, not all of them,
like
but
here,
that
this
one right there, you can see there are illustrations for each character state. Now, so I use that all the time, right? Short of populating each cell. The question about the labels, if you double click on that, you should open up a larger so not rather than just hovering, I think if you double click, it should open up that character dialogue exactly. And then you see media, that's right. And then you double click on one of those, and depending on who coded this, you would see it, you have this really nice function in MorphoBank, which I really like in general, with a few qualifications that I'm slowly getting to, hopefully not painfully. But you double click on one of those images, maybe someone put a label on there. And the default text would show up and saying 0, big, 1, something like that. Now, what would often happen in the old version, well, the current version, and that's I'm leading now finally to my question, is that if the bits of that image were close to each other, the interface wouldn't let you accurately put the little what do you call it, like in a cartoon,
pointer?
when you want to add dialogue. The arrow plus the box where you have the text.
Yeah.
To
It was a bit
annotate.
clunky. Yeah, so that's my
But
question.
you
Basically,
can even zoom in now. So like if there are two things really close to each other, you can even zoom in on the media image to annotate exactly where you'd like. But I haven't
great.
seen Tanya, I don't think I've seen any problems about.
Right now, if I double click on this, it doesn't expand. You have hit on a known bug. So we're working
Okay.
on that. So somebody else has
But
noticed it.
those labels are hugely helpful
Yeah.
to me. I
Mhm.
really depend on, and it's a really good functionality. I mean, they're all the projects that are in MorphoBank in the archive that are now permanent part of the permanent record
Mhm.
have not all of them, but many of them have these labels, which really I think are quite important. Because otherwise you have to go into like Inkscape or Illustrator and
Mhm.
it is possible to make two-dimensional images.
Right.
So anyways, that's my question. I'll stop talking. Thanks for that.
Mhm. Yeah. So thank you for pointing that out again, because somebody has mentioned it. You've mentioned it now. So we know that this is a high priority for us to address during the beta testing phase before we push it out into production. So thanks for that. Can't find my chat window. Here it is. Okay. So 15 new questions.
It's just a discussion on the gap or the dash
Creating
versus a question
labels.
mark.
Okay. You want to take that, Brooke?
So yeah, so I mean you
I think dash is missing, question mark is unknown.
yeah, and I mean you as a when you're coding the matrix, you can define it however you want. There is an option to add notes. So when you have a character state, you can like if it's a question mark, you can add notes to explain what that means to be more clear.
Right. So you can say, what does question mark mean and what does dash
mean.
Mhm.
Mhm.
Okay. That was the old site. Okay, so I did want to go to another project to show you folios, because the last project didn't have folios. This is another media heavy, but not as heavy as the previous one, project. It has no matrices, but it does have a bunch of media files, and it was a characterization of a plant family. It has 124 media files, and if I look at it in the thumbnail version, I'm like, oh, those are kind of nice. I can look at like 100 of them at the time, but maybe I want to look at them in blocks that show the media grouped by species, and you can do that in folios. So for example, we have 8 folios, and I'm just going to pick one of them. This groups the images according to this criteria. And it doesn't have to be grouping by a species. It can also be grouping by a part. You can determine how you would like to group the images in your folio. So back to that, there is a permanent link that you can use to access the folio specifically. Looks like that's a bug that we need to work out, but the idea is that you can create groupings that we call folios to put images together if you have a whole lot of them and you would like to make it easier for the people visiting your data to look at them in related groups. So with that, we've got the overview almost completed. Maybe one last thing you may have noticed before I hand it over to Brooke is that because the NSF has supported this project for so many years, we started tracking quite some time ago which of the projects deposited were supported by the NSF so that we could report back. And so if you see this note, that means that we have gone through the paper if it was curated by us and looked if there was NSF funding, or if it was submitted by a community member and they picked that it was supported by the NSF, that's added. You'll also see authors' institutions. So we track also the institution of every author, and if an author has more than one institution, we associate more than one. So in this case, there was that many institutions. in this other project, there were fewer. And this allows us to reach out to institutions for potential memberships, because we can say your institution has this number of projects in MorphoBank. And that's one of the ways that you can browse all of the projects in the repository. You can look based on the university. And I think we have one by count. And then do it the other way. So right now, this isn't exactly what is in production. So you'll notice that Brooke said earlier, we have over 2,000 published projects in production. So that's the live site. Since we're still in beta testing, it doesn't exactly copy all of the data in the production site. So it's a little less. I'll stop here, see if there are any more questions about browsing or viewing data. And then I'll stop sharing and hand it over to Brooke.
Okay. If there are no more questions, let me go ahead and share my screen. So I had planned a little short break. So we'll have a 3-minute break right now and maybe meet up at 45. But in the meantime, if you're not interested in taking a break, you can check out our beta sites. And if you have any feedback, if you could provide it within the next couple of days to curator@morphobank.org. And so the beta site's still going through testing, and there may be some bugs. So we're happy to get any opinions or feedback that you can provide. So we'll meet back about 2 or 3 minutes. Okay.
That's good.
I'll just leave this up on the screen for now.
back. At least I'm back. Okay, let's just look for signs of
Okay.
life elsewhere. If you are back, can you send a thumbs up?
Okay.
Yay.
So we'll go ahead and get started. Okay, so I'm going to go over a tutorial of the different components of MorphoBank and how you would add information and data yourself. So this includes things like taxonomic names, specimens, whether they're unvouchered, vouchered, media, matrices, how to do the phylogenetic analyses within MorphoBank, any documents that could be maybe matrices that have molecular data, or if you have associated morphometric data, collaborators, reviewer access, copyright status, and how to publish your project and citing the data. You can see my screen still, right? Okay. So here is morphobank.org. And this is not the beta site. And so what we're going to do when you're browsing and looking at the different projects on MorphoBank, you can access them without signing in. If you want to create a project, you will need to log in or register if it's your first time. So I am going to create a project. What I'm going to do is create a project for this paper right here. So we do curate data that is not in MorphoBank, and we add data to MorphoBank. So this is a paper on our list that has a morphological character matrix, but is currently not in MorphoBank. And so a way I can check that, I can just copy the title here, go to MorphoBank and do a search. And while that's searching, I will go through the paper. Let's see. So there's some trees on this paper, but there's also right here, so table 2 is a character data matrix. And so this character or this matrix is what I'm going to show how to upload. Okay, so back to MorphoBank, I did a search for that title and the paper is not in here. So if you have a paper that you've already published with a matrix and you'd like to add it to MorphoBank, you can easily do that. Either register or log in. I'm going to go ahead and log in. Okay, so you'll be brought to this My Projects screen, and this shows how many projects you have in MorphoBank, how many of those projects have been published, and how many are unpublished. And they're sorted by unpublished is on the top, and you can scroll down and see the ones that you've published below. And so all of these are, some of this is my data, but for the most part, this is data I have added into MorphoBank. I'm going to go ahead and show you how to create a new project. You can do that by clicking right here. Okay, so now there's some basic things that are required, and you'll see in orange next to it if it's required or not. So the title, and this is the title of your project, which can be different than the publication. It's up to you. So if we scroll down a bit, there's journal publication information, and that asks for the article title. For this purpose, since I'm adding data that is not mine and from an already published project, I'm just going to use the title that the paper has. And so we can paste that in. Is the project supported by NSF? I can do a quick, sorry for scrolling too fast. I'm just going to go down to hopefully acknowledgements, unless I've already passed them up. Okay, so typically we look for any, when we're curating, we look for any NSF support and we make sure to report that. So this one does have US National Science Foundation support. So all you need is the title, if it has National Science Foundation support, and that should be all you need. This article has already been published. And once I said it was already published, then you need to enter this information. So the journal title, the abstract, information about the authors, how it was published. If you're starting a new project, you can just say article is in prep or review, and none of this information is required. So for this purpose, because this paper was already published, what I can do is use the DOI to pull information. So I'm going to switch. This journal article is published. This information about if the journal article is published is different than if you want to publish it on MorphoBank. You would leave this as unpublished until you are ready to make any information about your project publicly available. Once you hit publish, it saves it and you cannot edit the project after that. So this step right here, if you're on the screen, do not hit this until you're absolutely done. But there is another way you can publish your MorphoBank project that I'll go over as well. And right now I'm going to go ahead and add the article DOI. I'll hit retrieve. Ideally it works. Do I need to have DOI in there?
No, it should work that way.
It should work just that, yeah. Yeah. There we go.
There it is.
Okay, it just took some time.
It's just tired.
Okay, so based on the article DOI, it was able to pull journal information, the page numbers, volume number, the link to the published article. So that way it ties the published article with the data that's in MorphoBank. And then it already adds in the authors. And what I'm going to do is go ahead and add the abstract. Just copying this, I'll add this in. If you have a published paper, we would like you to add the abstract here. If you are actively working on a project, you don't have to have your finalized abstract in yet. You can wait until you're ready to publish the MorphoBank project, and you can wait until after your paper is already published in a journal. Okay, so here, this is just the basic how to create a new project. We have the project title. Is it supported by NSF? I'm skipping a media image for now. I'll add those later. Here is where you can add information about peer review. So this section right here, if you are submitting your data and your paper for peer review, you can get a reviewer login and you would create a password. And then you would give the journal and the editor the password and the link to your project. And through that password, they can access the data. We're leaving this project unpublished on MorphoBank right now. And then here is all the journal publication information. And once I hit save, it will create a new project. But I'm going to pause and see if we have any questions for this initial creation page.
There are some comments in the chat, but they're not related to the project creation.
Okay, so I'm going to go ahead and hit save. And we'll give it a little bit to save it.
Now you can
And
see
the
why
beta
we want to
version, this
move
should
to
be
the new
quicker.
version.
Yeah,
right.
Yeah, the beta version, this should be automatic.
The fallback is to go and do this in beta.
Yeah.
maybe to add a little more context, is that the software for MorphoBank is about 20 years old, maybe more. And so it's time for the update to happen. But now it's completed, the saving, and we can
Now it's
continue.
completed. Yes. It only took maybe 2 minutes, or maybe a minute. I don't know. It wasn't that long, but, but for modern internet, we're usually faster. Okay, so it says at the top, successfully created a new project, and you'll see it here. So this project is now numbered 6047. So this is the official MorphoBank project number. It has the information right here about the authors of the paper, the year it was published, the title of the paper, the journal it was in, along with volume, number, and page numbers. And so typically I just double check to make sure that all that information is correct. And you can always go to this tab right here. It says edit project information. So if you click on that right there, then you could update any of this. And so we see that the abstract that I pasted in is now there. Because I clicked that it was supported by NSF, that little icon shows up. Down here, we have the article DOI, and we have read the article. And if you click this, it should redirect us, yep, to the DOI and the page where you can access the article. And there'll be a project permalink once we publish this, but it does tell us what this information is ahead of time. And so this is useful if you are submitting your paper for peer review and you haven't published it yet, you can still refer to your data in MorphoBank with this information. And so this is just ending with your project number right here. So for this one, this is 6047, which matches up to the one up here. Okay, so here's the basics. We have a new project. So the next thing I'm going to go ahead and do, and if anyone has any questions, please feel free to put it in chat for now. Okay. Let's see if I can open. Yeah, perfect. Okay, so for this paper, okay, we have a matrix with, it looks like 51 characters and not that many species. So I'm going to go through, I do have a file that has, I have a Nexus file that has this matrix, but I'm going to show you how to manually enter the information. So say you want to start off, what I'd suggest is start off with your taxa. What is the family or group of species that you're working with? So I'm going to go here to add new taxon. And for this, let's do the outgroup. So genus, species, you have the option to check if it's extinct or not. And you also, there's additional fields that are optional. You can add the scientific name of who discovered the species along with the year, add parentheses for the author or not, and any kind of notes. And so these notes are related to the taxa. You can change the access. So this is, if it's just me and I'm maybe don't say I start a new project and I have students that I'm sharing the project with, maybe I don't want the students to edit the taxon, but I want them to be able to see and work on the project. I could change this to just owner, or if I want anyone, say I want the students or collaborators to work on this taxon and add or edit it, then they can do that. So say I have a co-author that has the scientific author name and information, then I'd want to make sure that anyone can edit this so that they can add that information in and not me. I'm gonna go ahead and click save. And that is now saved. There's a confirmation at the top, and I can click here back to list and it will show me the list of the taxa added. So right now we just have one taxa. So I can add, if I wanted to, I could add each of these individually. Another option I can do is upload taxon batch. And so this would be if I have an Excel spreadsheet or a CSV that has information on the taxa being used. And we even have a sample file, so you can click on this and maybe add in your own taxa. For this one, it's not that many, so I could add it in individually. There's also the option to validate the taxa at PBBB. Okay. You can see this window, right? Even though it's not loaded. Okay. So usually this would be multiple taxa listed here, but since I've only answered one, there's just one listed. I'm going to click the checkbox and I can search PBBB. And sometimes there are multiple options that come up. For this one, I know that this is a bivalve, and so I'll double check. So PBBB found that, okay, we have the genus name is correct. There's family, order information, class, bivalves, phylum, mollusca. And so this looks like it's the correct entry. Sometimes, I mean, maybe a bacteria would pop up or something. Make sure that when you do this, that it is the right information and the right taxonomic details. And so I'm going to click a check right here and import. Okay, so that has been added. And so now if I click here and I click edit on this taxa, so I only entered the genus and species information, but now if you scroll down, there's phylum information, class information, order, family. If I wanted to add in subfamily, suborder, any of these different things, I could add it. I could hand add it. Or like I mentioned, there's a CSV file. So you can have all of this information in your own file and then upload it for all the taxa at once. Okay, so that is the taxa tab. I'll move on. So for specimens, so say you have a taxa and you have maybe 10 different specimens for that taxa. Let's go ahead and add a new specimen. So you can only add specimens for taxa that are listed. So when I go here, I can add a new taxon or I can search. What were we working with? Okay. So as I start typing the name, the different options for whatever taxa I had entered pop up on. So right now we just have one thing. I'm going to go ahead and click that one. But I can enter, is this an unvouchered specimen? If it's unvouchered and it's something I'm using, then there's a lot less details for it. If it's vouchered, which is our preferred use method, then you'd have institutional code. So I don't have the information for this one, but it could be something like AMNH, UF for University of Florida. There can be a collection code. So IP for invertebrate paleontology. It depends on the museum, but you would have that information along with the catalog number. Sometimes there's an occurrence ID if you want to enter that and any kind of notes. There's also the access information. So anyone in your project can edit it or just the owner can edit it. For this, I'm not going to save it because I don't have specimen information for this taxa. And you don't necessarily need specimen information for the taxa. If you have a media image, then you would be required to say what taxon does it belong to and give information about the specimen for the media. Okay, so now I'm going to go ahead and go to matrices. And so for this, we can create a matrix from scratch. And for this, I'm just going to put a title of test. You can add any kind of notes that you want. And you can set what the OTU is. For this, we have species that are listed. Yeah, so the default is genus. You can leave it as that, or you can update it to what you're working with. For dinosaur stuff, most of that is at subfamily or family level. And then there's options of publishing status. Are you publishing this matrix when the project is published, or are you not? And so typically, yes, you would want to publish the matrix and you publish the project. I'm going to go ahead and create this. So this is just an empty matrix right now. We have one taxa because that's all I've added. But oh, you know what? I might need to change. Okay, so this is a pop-up in a new window. I believe, Tanya, if you can, for the new version, it will pop up in a new tab.
That's right.
Right. Okay. Yeah. For right now, this version is a whole new window. And what you'll see is information on the taxa. And usually there's more taxa than this one right here. But you can add character information here. So for example, let's add a new character. Oh, I might need to share my screen differently.
It's okay. I can
So
see the pop-up window, Brooke.
Okay. Okay. So what I'm going to do is from that paper, in an appendix for the paper that this is from, they have a list of characters and character information. And so the first character, for example, is called size. The character name is size, and there's nothing else. So I'll add that new character. And so as you can see now, the name of the character is size. I want to add states. So in the paper, state 0 was called, and I'm just copying and pasting this, it's called tiny width of less than 1 centimeter. There is more than one state, there's a few. So the next one, coded as 1, is small with 1 to 3. We have another state, medium, and there's a 4th state for large. Okay, so now I have created a new character. I have the name and I have the different state options.
I'm gonna
You can add.
I'm gonna pop in here and just say that in the beta version, when you create a new character, there are 2 states created by default. So that, and then you have to fill in what they are, but it's a little bit of a time save if you're creating a matrix from scratch.
You can add any notes. You can say if it's ordered or unordered. You can add media associated with this character. Any kind of comments can be added, and it will tell you if you're collaborating with multiple people, it will tell you the user that added any comments. You can add citations, and so for characters, I think the citations is really useful. Say that these size categories, the 4 different options for the states, if that was from another paper and you just decided to use the exact same characters from another paper, you could add citations and reference information. And you could say, this is from whatever paper, here's the information, it was found on page number, and then add notes. You can add partitions and there'll be a change log. And we can go into this more once I add the full matrix. But for now, we'll just have this one character. So we just have one character and one taxa I've showed you how to add. And so now it's showing up here. There's size, there's no notes. I didn't add any notes. But then there's the states and the media option. And so right here, now we're back to the matrix, and here's the taxa. And for size, there's a question mark. Now I can look at the matrix from this paper and it says for character 1, the species is coded as a 2. Oh, here we go. We were asking about the NPA and the dash and the question mark. So here are the other options. I think the default is question mark. And so for this one, I'm going to click 2 for medium, and it automatically changed it from question mark to a 2. I'll click done. And so now it shows up that this taxa for the character size, the state is medium, and this is set up right now so you could see an image if there was an image shown. I can also change it. So right now up here it says show name plus image. I can say show names and numbers. Oh, could be. Okay, that's a bug because that should show just a 2. Okay, so then, oh, go ahead, Tanya.
There is one question in the chat. Can you talk a bit more about the commenting function? I had difficulties of finding comments from other people in my collaborative matrix.
Okay, so I'm just gonna add in a comments. And I'll save it and it, test on this day. Maybe I can say something like added for character states. And so I'm saying what I'm doing here. And so maybe you have a collaborator and they updated this in some way. They can go to this comment section and explain what they did. Tanya, do you know of any other use cases?
I think if we go to the character itself and then double click on that, you can put notes or comments there by clicking on the add new. And then now so you can add a comment on many different things. What Brooke commented on earlier was on, I think, the score, like the scores. And right now she's commenting on the character.
Okay.
So you'll see the ability or this tab for comments in many different places. And so people can comment on many different things in particular, as opposed to just adding a general comment. So it might be that people in the collaborative project have found the comment section in one place and are putting all the comments there. I'm not exactly sure how they're using it, but that might be why the comments are distributed. Yeah, Emmanuel, please go ahead and unmute to follow up.
Yeah, just to give a bit more context, thank you. So at the bottom right, you have this mark comments as unread. And then when you actually go to the main page where you can see the matrix of the project, so the thing is, when people are working at different times on the matrix and people make comments on the character or character state of how they think it should be changed to make it more understandable, then be able to quickly find all the characters who have such comments so that I can work on them without having to go through every single character to look if they have comments or not. And I haven't really found a way to do this yet.
That is a good question. I don't know right now whether we have the functionality to do that, but we'll certainly write it down so that in the new version we can build something like that in. And while Brooke is demonstrating more functions, I'll have a look and see if I can find something. Okay, yeah, thank you.
Okay, are there any other questions about just the basics of how you would add a character and character states? And I'll show you another version. I'm gonna upload the whole matrix next. Okay, So I'm going to go ahead and add a new matrix to this project. So what I had just done was show you how to create a matrix from scratch. So that's no previous information, or maybe you have, you know, some of it half done in an Excel spreadsheet and you want to just upload some information. You can do it from scratch and build your matrix however you want. There's also create a matrix by uploading an existing file. So for this, we already, since this is a published project and or a published paper, and I do have the Nexus file for it. Let me see. So I'm going to give it a title. Okay, so I need the matrix. Okay, so I already have the matrix, and it is the matrix that is shown right here. So I have a Nexus file that looks like table 2 or has all the information of table 2. And let me actually show you what that Nexus file looks like. Okay, so this is the Nexus file that I'm going to upload. And as you can see here, it has information, what the title is, the number of taxa. So there are 20 taxa, and it gives the taxa labels, so the different species. And then here we have the character matrix. And this looks similar to the table 2 that was from the paper, where all the information is listed out. And at the bottom, so here is, it could be, this could be a Mr. Bayes block, this could be Mesquite, this could be tree files that are emitted on here. MorphoBank should accept anything below the matrix because usually there's other information associated with it. So I'll go back now that you've seen the matrix. So that matrix, I just attached it right here. There's a choose file option, and it is a Nexus file that I've added. So I'm going to click create. Oh, okay. Let's see. Does it not, if there's no characters at all, Tanya, it's not, it won't let you, nope, even add it yourself. Okay. So there was, so that other file that I showed you, the Nexus file, did not have character information. And so this at the top, where the character names would be, is just 1, 2, 3, and hold on. One of my
Switching technology.
Apologies for the technical difficulties. Okay. So because we don't have character names listed here, MorphoBank will not accept it. And so that is one of the things we've been working on, and we encourage you when you're publishing your information or when you're publishing your data that you have all the information. And so the information is not in table 2, it's not in that Nexus file, but it is down here in the appendix. And right now, Tanya, can you answer any questions while I get the file that we'll upload?
Yes. So Emmanuel, I was able to find something that might help. So let me temporarily share my screen. Let's see if this works. So this is in the new version. It's also present in the current version. So if I go to characters, oops, I see all of the characters, and that was not it. It was search. So it's this search magnifying glass right here. You can search for characters, taxa, and cells. And I found one that might be useful that exists already. And that is you can search for characters with unread comments. So in this particular matrix, there aren't any, but you might try this in yours and see if that works.
I think I did try this, and there was a problem with it. Maybe once somebody actually read a comment, it turned the unread to read automatically. But maybe that was not the person who actually wanted to
read it.
Well, not do the modifications that were suggested.
Right.
So then it cannot be found anymore. So maybe just an option to search for characters with unread and read comments, with comments in general.
Just
that
with
might
comments
be,
in general.
yes,
Okay, that's a good idea.
that would be useful. And also what we thought, like in the beginning, in the stats of the matrix, I think you can see how many comments there are. And if there could be a link there to click on that number with the comments, that brings you into a kind of a list of all the characters with comments.
So let's just go here.
No, I think it was in the project overview, somewhere below. Or you, there is like who made.
I'm going to scroll
character
slowly.
Notes.
Maybe this. Cell notes or character notes.
Yeah, this might also be interesting. If you could then click on the number, that you would just get the list of all the notes of Maureen O'Leary in this case.
Okay.
I don't know, something that would link these numbers to the actual data they are connected to would be great, I think.
Okay, I see what you're explaining and like what the use case is. So something that we could then take to the software developers and explain so that they could implement it. So again, something to go on our wish list. I can understand how this could be very, very useful for a collaborative project. So thank you for bringing that up.
Yes, sure. Thank you for listening.
Yeah. I'm going to stop sharing and see where Brooke's at.
Yes. Okay, I am back and I have I have a Nexus file that MorphoBank will accept because it has all of the character state information. Okay, so what I did is we haven't made this accessible yet to the community, but we haven't a way that we're using AI to take information from papers. So for example, I have a Nexus file that has the matrix, but there is no character information. And so the Nexus file has the information from this table, but in the paper, there's an appendix that has the character names and descriptions. And so we have a way to use AI to pull this appendix information and add it to the Nexus file here. And so I did that. And now let me go back. So I'm creating a new matrix. I have a title and you can call this matrix whatever you want. If you're having multiple matrices, make sure that you're giving it a detailed title so you can tell the difference between them. So I went ahead, I added this Nexus file here. I hit create, and now that those character states are in there, MorphoBank accepts it. And it found 51 characters, which is good because in this appendix for the paper, there were 51 characters. And it has, let's see, scroll down. So there are a couple issues for this particular Nexus file. If there's any kind of problems, the information appears in red. So for this first character and character state information, this is the one I had manually added in the other example or its size, and then the 4 different states. The AI, the AI tool I used, for some reason kept the 0, 1, 2, 3, but not for everything. And so for this, I can either go in here and fix it now, or I could fix it later. But for the example fix, you can edit anything right in this upload window. You can save it. There we go. Okay, so now those are removed, but as you can see, they're in almost all the other characters. And so I'll go update that later. I would not publish this matrix with that in there. But it did find, okay, so for example, state 3. So in the matrix, and somewhere in this table for character 14, there is a state 3. And so character 14, I don't know if you can see where my cursor is. I'm gonna go down.
see your
Okay, so
cursor.
there's a state 3 right here. But that state 3 for character 14, here's character 14. Okay, there is no state 3 listed. And so if you were working on your own project, you would know if there should be a state 3 or not. For this, I will just say okay, and then so now this shows up here. Although MorphoBank had some issues with the degrees, I can update that later. But now it's no longer appearing in red as an issue. And you need to fix, when you're uploading a matrix, you'll need to fix all the problems that pop up in red. Otherwise, it won't let you move on. And so for this, so let's check character 50. Character 50, there is no state 2 listed.
I'll pop in here to say that this is a really nice check when you're uploading your own data to see, did I forget to define state 2 or state 3, or is my
Or
scoring
maybe it was
correct?
a typo. Yeah, it was,
Right.
I mean, the scoring for, what was it, 14? The scoring for 3, maybe that was supposed to be a 2.
Right. So it could be either of those things. And if it's your own data, you probably know right away, oh, this is what it should be.
Mhm.
When we're
And
curating other people's data, it's impossible for us to tell which of those it is. And so what Brooke just did is what we end up doing.
And if this is your own data, this is a great check. I mean, before you run your analyses and have your tree that you publish, it would be awesome to know if there was an issue like this, right? Okay, so now that there are no more red marks in here, even though there are a lot of issues in this matrix, I would not publish this as is. I would fix that, but there's nothing red that's popping up. So I can click next and it will save that. And now it's saying it found 20 taxa in my matrix, and I can go through and make sure that there's no taxa missing. This would be something I can compare to the original publication, or for my own, if it was my own work, I could check that. I'll hit save. And now we have that matrix in MorphoBank. So here was the test one I did where we just had one taxa, I had added one character, and I scored that one taxa. Here is the one I just uploaded. And so there's 20 taxa and 51 characters and over 1,000 scoring. So I'm going to click edit matrix. Then I will need to switch. Okay, so here is that matrix, and what you're probably used to seeing is this. And so here's your taxa, here are all the scorings, and we have information at the top. Which you can do here. We can also switch it so you can see images or numbers and images, and you can link media. I'm gonna go ahead. I'm gonna show you how to add media, and then we'll go back to this. Okay, so media. Let's say, okay, so this, I might have a lot of media. I can either add a single image, and I would upload a file, and I would enter information. If side is present, or if there's not a side, I'd link it to a specimen. I would have a view. I can add information about the copyright, media URL, and then you can publish it. I'm gonna go back because I don't have media. I can search for media. Okay, so let's import iDigBio media. So here is all the 20 different taxa. They can select all, and let's search iDigBio to see if there's any images of these taxa.
And it's normal for it to take this long, so don't be discouraged. I think it's a little bit faster in the new version.
Okay, so here are some of the results, and it will show a little clip of what the media looks like. For this case, I'll just scroll through. We'll just go with the top one. So you can import it, and you can import all of them. Say I want to import, I'll just go ahead and import all of them. I can select one to use as a taxon exemplar too. And so I will import. I selected 3 images to import.
The reason you might want to select something as a taxon exemplar is that then it shows up in that square in the
matrix. Okay. So I can add, what you need to do next is curate the information and make sure it uploads right. So for this one, I would want to, I think I'm going to need to add a new view. For this, I mean, I could say they're shells, so I could say external valve, internal left, right valve. That one's super tiny. For these, just to keep it simple, I'm gonna just say like whole shell. But for your research, you can make this as specific as you want. Okay, and so now, because I picked a view, or I named a view, I said all these 3 images were whole shell, and so it shows up in the information here. And I'm gonna go ahead and click all 3 and click release selected media. Media are released, so now they are available. And I'm trying to get it to show up. If you can see, there's information here about copyright permission. I'm gonna go ahead and click on this image so we can see more information about what is listed here. Okay, so this specimen, it's pulling information from iDigBio. So this one must have been listed as unvouchered. The copyright holder is PRI. There's some default information about the copyright because I pulled it from iDigBio. And there's information for the URL of the media, some information. So this is automatically generated if I'm pulling media from iDigBio or from EOL. And so automatically pulled from iDigBio and some more information. I don't think I need to save any of this, but say you were to upload your own information, you could pick a side, you could pick copyright information. If it's vouchered, you would say it was vouchered and what museum it was a part of. So this one's from PRI, so it probably is vouchered, but the information in iDigBio does not give more information. So we're just showing whatever iDigBio had. Now, when I go back to Matrix. Oh, that was my test one. Let me go back to the whole matrix. So right here, I had clicked to use it as a taxon exemplar for one of the media. And so that is what is showing up right here. This image, and it's a little thumbnail that goes with the taxon. Say that I wanted to use that same media to illustrate the state. Let's see, I can.
Yeah.
And this should all be faster on our new upcoming version of MorphoBank. Okay, there, there it is. So it's showing me the media associated with this taxon. And so say that I want to use this. I'm gonna add it. And I guess there is a scale bar in here, so that's good. So I think this was the character with size, and there's a scale bar. So maybe I'm showing off that this specific individual meets that size range. And I'm adding that as a character or as a media for that character. And if you have for a specific taxa, you could have maybe 15 different specimens. And for each of those specimens, different photos for each specimen to show off different things. And you can add all of that media, connect the media to the specimen, connect that specimen to the taxon, and then pick what images you want to show here. Well, this is saving. Does anyone have any questions?
There is one question, I guess specifically about this character, but you can clarify kind of in general. It says for the size, are you always using adult specimens?
That would be up to you. I mean, yeah, ideally, yes, but so say you're working with juveniles, a different character state or a different character name could be juvenile size or max. And I mean, this for size could be maximum adult size to be more specific. And that's something where there's notes. So when you create the character, say you just called it size, in the note section, you can say, this reflects the maximum adult size or the average adult size based on 25 individuals measured. You can add information to support how you determined the different characters and the different character states.
We have a question about in the matrix editor. So I see your question, Malena. We're going to take it later during the next section, but there is one now for, let me just find it. Best practices in MorphoBank. This is one that's good in general. If I have several articles and in each one of them I perform phylogenetic analysis, each one of them should have the corresponding matrix, or just one matrix would be updated as new data is added? Go
You would
ahead.
have multiple matrices. And so that happens a lot. So say this matrix that I'm working with, this was done in 2015. Now it's 2025. If I want to use this exact same data to either reanalyze it in a new way, I would create a new MorphoBank project and duplicate the matrix. And I could keep it exactly the same. And maybe I have new trees and I've done a new type of analysis. Or I can even take this, we duplicate the matrix and say we're adding one more taxa. Maybe there's a new species you want to add in. So we would be using this matrix from this paper, but it's new research. So you duplicate it and add in the new specimen. Or maybe you want to go in and change the characters. Maybe the size isn't accurate. Maybe you want to say adult size, juvenile size, maximum size, minimum size. I don't know. You would have multiple matrices and multiple projects depending on the scope of the research.
Thanks, Brooke. And then one question that I've answered in the chat, but I'll just say it out loud, is that media have to be linked to a specimen so that you can upload it. You cannot just upload a bare image. And they can be unvouchered specimens if you don't have that information, or it's not vouchered. It has saved?
It has saved.
Okay. We've killed enough time that it has saved.
So now that picture that I picked is now in showing for character 4, and I believe, okay, so here's a different taxa, character 4, I could upload a different image to show up here. And you can even add multiple images. So for this one, I don't have multiple images, but say I have multiple specimens that I want to show off to show that they're all within whatever the scale bar is here. I think, so for this one, let's see, character size medium, 3 to 6 centimeters. So say I want to have 10 pictures here that prove that these shells are all 3 to 6 centimeters. I could have that and you can scroll through it. There's also, now that this picture's popped up more, there's the annotation features. So for this is size, maybe I want, maybe I want to do something like show size of something. Or size of the valve. Okay, I'm having trouble getting it the right way. But you can point to different features, you can call out different things. Oh, there we go. So you can use lines, you can use squares. But different call out features. Yeah. Okay.
Yeah. The software is slightly different in beta, so it won't look exactly like this because we had to use a whole new package for it, but the ability to point, to draw a rectangle and to draw a polygon will be there as well.
And now here's the 3 label. It says 3 labels. So if I click on that, it's possibly saved it. Yeah, and there's all the messy ones. and you can edit that. And so me as a curator adding this project, I would not add any detail like this because I was not the authors and didn't decide how this is, how the size is determined. You can also, so I'm going to switch to a different version or a different way to view it. There are partitions. There we go. Add new, let's maybe just say we want to call it outgroup. And say I just want to look at the outgroup taxa. So I just pulled those 2 over to a partition. And I mean, I can also specify characters. So let's say I just want to look at the tooth orientation for these outgroups. Now I have specific information here for the partition. I'm going to hit done. And then I believe, there we go. So now show partition and matrix editor just outgroup. So here are the 2 taxa I selected and the characters. And you can make as many partitions as you want. So Teeth. So say I want just the teeth as characters, but I want all the taxa. That's also an option. Oh, might have messed up that part.
Okay. Okay. It's okay.
No partition. Okay, I don't know why that teeth one isn't working.
I don't know either.
But as you
in
can see in the
principle, it
background,
should.
there's, yes, so in the background, there's no partitions. And then when I click to outgroup, oh,
Mhm.
now MorphoBank's happy with me. Oh, no, it's the, okay, so this is the tooth one now it's showing,
Mhm.
but for everything. Maybe I'm telling it to do things too fast. And that could be it. Okay, so that is what I have to show you for the basics of what you can do with the matrix editor. And there are a ton of different things that you can do with this.
So we have another.
Actually, this might be a good time to go to the question from Milena, which was, if you could unmute yourself and say exactly what you're trying to do. It was something about doing a batch of characters, of editing.
Hello.
Yes, hello.
Uh, I find like a good practice to order the characters by bone, part of the bone, because I work with appendicular characters and sometimes they are very far away from each other in matrix, in all matrix.
Mhm.
And for that, I have to put the same in a batch of character, and I have to edit one by one by one. And also a lot of time I like to put the original the original reference in the notes, and it's the same reference, so I have to edit a lot of characters for the same thing, and I have to edit them one by one by one. So I was
Mhm.
asking if I can like change a lot of characters at the same time.
And are you changing the order of the characters? Because you're saying they're like,
No,
might
no,
be
no,
far away.
the wording of the characters.
So if you're editing the wording of the characters, you do need to click on each character
Yes.
and change them individually.
Yes.
Besides that initial upload screen, so when I uploaded this matrix that we already had and everything showed up at once and you could edit the different character state information, that the upload spot is the only spot I know of that shows everything at once where you can edit each character. Otherwise, you would need to go in, edit the matrix. Although
I'm trying to think of what that might look like for you ideally. Is it like if we imagine an Excel spreadsheet where you could just go from one character to the next and edit, kind of like the upload screen, or like if
So
you
like-
had to design it, what would it look like?
Well, there, for example, since I want to put the quote in the notes, the original matrix I take the character from, I will have to go one by one, changing the note.
and add the notes here and then save it and then go to the next character.
Yes.
But maybe 50 characters are all taken from another paper and you just need to paste in the same note information.
Exactly.
Okay.
So we don't have a capability right now for that.
So what I'm doing in the chat right now is I'm trying to write down suggestions as clearly as I can capture them. And if that's incorrect, please, add something onto what I put in the chat so that we try and capture what the desired features are. So at least we know what you'd like, and then we can prioritize them for
Thank
working
you.
on. Let me see. I think there was one question on how you run an analysis in MorphoBank.
I was muted. Okay, I was muted. Okay, so now that I have this Nexus file here, and I can go down to this, click right here, build a tree from your data. And you can only do this before the project is published. So once you publish it, you won't be able to access the matrix editor. You won't be able to build a tree. But what you can do is request for us to duplicate your project. So then you could use it for future projects and additional research. But you click build a tree now. Right now, we just have PELP in this version. And so our other version will have TNT and Mr. Bays, but right now, we just have, let's see, job name. You can have however many settings. We have some default settings, and if you want to learn more about what the default is, we have information here and some different links. I'm just gonna keep it as all the defaults, but you can edit it. And with our Mr. Bays option that's coming, you can upload a Mr. Bays block. So you can be really specific about all the different features that all the different settings that you want for your analysis. Authorization error. I might need to test this on the beta version because the beta version was working for this. So our new version will have this. I don't know why it's not running right now.
Yeah,
But
I don't
ideally
know
you
either.
click run and
Right.
down right here, so that says previous runs, there's none. The second I click run, ideally it will say queued and will have information. It'll have the job name, test information, and when it ran, and it will give you updates. It's run on Cypress, so it's external from here. But I mean, for something not very big, it will be done within a couple minutes. If you have more complicated things, it could take more time. I mean, if it's taking you, if you check back the next day and it's not done, please send us an email because then there could be a problem. And I don't know why it's not running here, what the error is.
Yeah, I don't know either. Sometimes, so we send the information from MorphoBank to Cypress, and sometimes that pipeline, for whatever reason, there's something up with it. And it could be something on our end, it could be something on the Cypress end, and troubleshooting that takes some time sometimes, but that is the process to get through it. So I think Brooke will try on.
Yeah, I'm gonna try on our new
beta.
version. So I know I had some things run, just trying to find my project. Where did my project go?
So while Brooke is searching, I'm going to read another question.
Okay.
What if you want to use an existing matrix from a published project? Can you reuse it as is, or do you need to create a new one with updated character descriptions? So there's two ways. If you do a reanalysis with exactly that same matrix and you publish a paper with it, then we suggest you still upload that matrix with your publication. Sorry, Brooke, you've got a little bit of background going on right now. Thanks. So that way, people reading your paper will know exactly which matrix, and they can find it associated directly with your publication instead of having to backtrack one. And the suggestion that came in the chat earlier of being able to link projects to show kind of the history or the provenance of the matrix is a really good one. We haven't implemented that yet, but that would also be useful. Secondly, there's the chance where you want to reuse an existing matrix from a published project, but you want to add maybe another taxon or two, or you want to add another character, or you want to rescore or update the characters. Then you really need to create a new project and a new matrix. And an easy way to do this is, there are two ways. One, you can ask to duplicate the project that has that matrix that you want to use, and then you're just going to update all of the associated reference information so that it's for your study. The other way to do it is to download that matrix from the published project and then upload it into your new project and make the modifications from there. I hope that answers your question. Okay, back to Brooke, maybe something has happened.
Okay,
Yes.
so you're still troubleshooting analyses, but I switched over to our new version. Just to show you what this looks like in our new version. So we'll have tree analysis at Cypress, which will be PAL bar Mr. Bayes, and then we have a TNT option. And so what this would look like once you run it, the second you click run, it will be put into something that's called queue, and it will start working on it. Right now, we're still having some errors come up, so this hasn't run yet, but there were two that I had previously run. So I had run a PAL and a Mr. Bayes run the same data, just two different options, and it's completed, and it will give you updates whether it's queued or still in process or completed. If it's completed, you can download the tree file. For this one, there were errors, and so it's telling me that it gave me results, but there are errors, and so I'll need to go in and fix that. And it should tell you what may have gone wrong in the process. But ideally, once this is all fixed up, you run it, it has we'll go through and ideally a short amount of time to say it's completed, and then you can click download and you will get a tree file. And the tree file will also have information on the original matrix used and the parameters that were set. If you get a sneak peek at the new version, like for Mr. Bayes, if I were to add information, I could either say that there is a Mr. or does it contain? Let's see, I can enter a Mr. Bayes block. And so for Mr. Bayes, you can add whatever parameters you would like. These analyses do not accept molecular data right now. So in our current version, it is just morphological data. So everything that this matrix upload option or creating a matrix takes is morphological characters. If you have a combination, molecular and morphological matrix, you can add that to documents. So that's somewhere you can add it. And if you want an analysis that has combined data, you will not be able to do it through MorphoBank. You'll have to do it on your own. Okay, so I think I've covered the vast majority of what you can do, or at least the initial setup of a project. You can manage members. So so you go to project overview, and on the right, there's manage members. You can add new members. And so this is adding students, collaborators, whoever else you want. I clicked add new member. It's taken a little bit of time, but I would then put in their email and I would select membership. This full membership option is can add anyone. I'll just, again, just add another version of myself. So I added an email. This email is not in the system. So it's not, that person is not yet a member of the MorphoBank community. So add their first name, their last name, and a little message, and they will get an email from MorphoBank to register. If that email was already in the system, then it wouldn't come up. It wouldn't have the first last name, but it would have what is the message you want to send, and then you can edit the membership type. A full member can edit anything. You can have an observer, so someone who cannot edit. So say you're teaching a class and want students to look at things, you can add an observer. A character annotator. So this is someone who can edit everything but characters and states. And then there's someone who can just edit bibliography. And so if you want a bunch of citations to be added here, that you can set a person to just do that. And then you would send the invitation. And so that is on the project overview page over on the column on the right. You can select certain member groups. You can request your project to be duplicated. So this is where if you're wanting to reuse the matrix, you can do that there. And then once everything looks good and you're ready to publish it, and you're done with your project, maybe it's already gone through peer review, the paper is in press, or even, after it's in press, you can go ahead and publish your project, and they'll ask you a couple questions. Is the research supported by NSF? Have you marked all of the taxa are as extinct or not extinct? If you haven't, there's an option where it will bring you to that. You verify that the data is
Brooke,
yours.
could you just click on the extinct option?
This
Because
one?
I don't think we had a chance to share it.
Oh, yeah.
Oh, it's doing this thing. It's just.
And so I don't know for these taxa, I think they're all living, but for example, if, a couple species, these species were not, you can move them over to extinct. And now they'll have the dagger added right there. And so I'm going to go back to the list, and now those three that I moved over have the dagger. Say that was a mistake, I can go to edit and I can unclick, is it extinct, and save it. And now the dagger is removed. Okay.
Okay, we have a couple of questions. Let me see. One is about the Cypress connection. So I've heard that Cypress will shut down in 2026. Is that true? And if so, is there a backup solution for when that happens? So it is true, Cypress is going to cease operations March 31st, 2026. Unfortunately, like very unfortunately, that's it's funding based, and that's why that's going to happen. The backup solution for now is the TNT analysis that we host on Phoenix servers directly from MorphoBank. We have looked into what it would cost to host additional analysis, and right now that is beyond what membership and Phoenix Bioinformatics support, and with the grant running out, can support. So we want to provide it, but we don't have right now the finances to support both PALP and the Mr. Bayes function. So Mr. Bayes and PALP will be available through the end of March next year, and then we'll be evaluating how we could, extend the TNT analysis that we'll be providing them.